Welcome to the deep dive. Today, we're jumping into the really interesting history of how we classify, well, everything alive on Earth.
That's right. We're looking at biological classification based on the materials you shared.
Yeah, and our mission is basically to pull out the key ideas, maybe some of the surprising changes in how we thought about this. We want to give you the big picture.
Exactly. How we went from, really simple groupings to the complex systems we have now.
Without getting totally lost in the weeds, hopefully. It's like the quick guide to organizing life.
It is, and it shows how our understanding evolves, moving from just intuition to really sophisticated science. It's quite a story.
Okay, so let's start at the beginning. It seems like humans have always sorted stuff.
Yeah.
Even before formal science.
Oh, absolutely.
The materials mentioned, these really early attempts were super practical. Like, is it food? Can I make clothes from it? Can I build shelter? Very need-based.
Totally utilitarian, driven by, immediate survival, but then curiosity kicks in.
Right, wanting to understand it on a deeper level.
Exactly.
Which sort of brings us to Aristotle. He made one of the first, maybe more scientific attempts.
Yeah, you could say that. He used simple physical traits, morphology, what things looked like.
So for plants, it was like trees, shrubs, herbs. Pretty straightforward.
Uh-huh. And for animals, even simpler. Does it have red blood or not?
What's interesting there is this shift, right? Away from just is it useful, towards looking for actual similarities between things.
Precisely. It was still basic, but it was a step towards using observable characteristics as the foundation.
Okay, fast forward a bit then to Linnaeus, the two-kingdom system. Plantae and Animalia.
Plants and animals. Simple.
Sounds easy enough. Everything's one or the other.
And it worked for a while, but then, technology happened, microscopes got better.
So we started seeing the tiny stuff.
Exactly, and our understanding of the microscopic world just exploded. And that simple plant versus animal thing, it started to look pretty inadequate.
Because it just lumped wildly different things together.
So,
Like, the sources mentioned it didn't separate eukaryote cells with a nucleus from prokaryote cells without one.
right. Huge difference. And it mixed up single-celled organisms with multicellular ones.
And even things like photosynthetic algae, which make their own food.
Yeah, they got put right next to fungi, which absorb food and are totally different in that respect.
So discovering organisms that just didn't fit neatly, maybe had traits of both, or neither, that really showed the cracks in the system.
It really did. We needed to look deeper than just, what something looked like on the outside or if it moved.
So the criteria had to evolve. We started looking inside the organisms.
Cell structure became important. What are the cell walls made of, if they have them?
How do they get nutrition?
Yeah.
Are they autotrophs, making their own food? Or heterotrophs, eating others?
Habitat, how they reproduce,
Yeah.
and crucially, their evolutionary connections, phylogeny.
Their family tree, basically.
Exactly. Considering all these different angles was absolutely essential to get a classification that actually meant something biologically. Just the big picture wasn't enough.
And all this need for better criteria, it leads us to R.H. Whitaker in 1969. His big idea, the five-kingdom classification.
Yes. Monera, Protista, fungi, plantae, and animalia.
This feels much closer to what many of us learned. And it wasn't just adding more boxes, was it? It was changing how we define the boxes.
It was a fundamental shift, yeah.
Yeah.
Based on those key criteria.
Right.
Cell structure, body organization, single-celled or multi-cell.
Right, mode of nutrition, reproduction.
And importantly, those phylogenetic evolutionary relationships.
It's interesting, the materials also nod towards the three-domain system and even a six-kingdom idea as sort of next steps.
Yeah, things you might cover in more advanced studies.
So even Whitaker's five kingdoms aren't the absolute final answer. Science keeps moving.
That's such a key point. It's always being refined. The three-domain system, for instance, digs even deeper into the prokaryotes Monera, recognizing huge differences within that group itself.
Okay, let's look at some of those big reshuffles then. Early on, things like bacteria, blue-green algae, fungi, they often got shoved under plants.
Just because they had a cell wall.
Yeah, which seems logical on a very superficial level, I guess.
But only superficially. When you look closer, it falls apart. You're putting prokaryotic bacteria, simple cells, no nucleus,
Right.
in the same group as complex eukaryotic plants with all their specialized internal parts.
And the way they get food is totally different, too. Plants making their own food with sunlight
Autotrophs.
versus fungi absorbing nutrients from outside.
Aerotrophs. And yeah, the cell walls themselves are different materials. Chitin in fungi, cellulose in plants.
So it became obvious fungi weren't just weird plants. They needed their own kingdom.
Exactly. That was a really important step, establishing kingdom fungi,
And the bacteria and blue-green algae, they got grouped into kingdom Monera.
because they're all prokaryotic. No nucleus.
Right. That tidied things up quite a bit cell-wise, separating them from all the eukaryotes, the organisms with a nucleus.
It was a major leap, recognizing that fundamental split in cell structure.
Then there's Protista. This one seems like maybe a bit of a grab bag for single-celled eukaryotes.
Huh. Yeah, sometimes called the junk drawer kingdom, though that's a bit unfair.
Okay.
But it did bring together organisms that were previously all over the place. Things like chlamydomonas, which used to be considered algae, with things like paramecium and amoeba, which were once seen as tiny animals.
So the unifying thing here is single-celled, but eukaryotic. They have a nucleus.
Exactly. Even if they differ in lots of other ways how they move, how they eat that shared cell structure became the key. It shows how new understanding makes us redraw the lines.
So the big picture is classification isn't set in stone. It changes as we learn more, especially about evolutionary relationships.
So it was related to whom?
Yeah. The goal is a system that reflects not just looks, but history, ancestry.
Precisely. Modern classification aims to be phylogenetic, reflecting the evolutionary tree of life. And that involves integrating tons of information.
Okay, great overview. So for the rest of this deep dive, let's focus on 3 kingdoms from Whitaker: Monera, Protista, and fungi.
Sounds good.
Let's start with Monera. Simplest cells, right?
Monera is unique. It's all bacteria, and they are just everywhere. The most abundant microorganisms.
Soil, water, extreme heat, extreme salt.
Inside us, on us, as parasites, you name it. And they have this incredible range of ways to get energy, metabolically speaking.
That's why they're so successful, isn't it? They've figured out how to live off almost anything.
Pretty much. They've exploited nearly every energy source available.
And they have different shapes, too. The materials show the coccosphere, bacillus rod, Vibrio comma, spirillum spiral.
Like microscopic pasta shapes.
Yeah. And those shapes aren't just random, right? They relate to how they live or move.
Often, yes. That visual diversity hints at the huge functional diversity. And within Monera, there's a really fascinating split, Archaebacteria.
The tough guys. The extremophiles.
Exactly. Living in places most life couldn't handle. Super salty lakes.
Halophiles.
Hot springs.
Thermoacidophiles.
Marshy areas with no oxygen.
Methanogens.
And the key difference is their cell wall structure. It's unique, lets them survive those harsh conditions.
Like the methanogens in cow guts making methane.
That's a perfect example. They're crucial for breaking down cellulose in ruminants and produce biogas.
Okay, so Archaebacteria are the extremophiles.
Yeah.
What about the more everyday bacteria, the eubacteria or true bacteria?
Right. Eubacteria typically have a rigid cell wall, and if they move, they often use a flagellum, that little whip-like tail.
And within eubacteria, there are some really important groups. Cyanobacteria, blue-green algae.
Very important, they're photosynthetic autotrophs. They have chlorophyll, similar to plants, and make their own food using sunlight.
They can be single cells, colonies, filaments, found all over water and land.
You sometimes see them forming those blooms in polluted water.
Right, the green scum. But some are really beneficial too, like nostoc and anabina. They have special cells. Heterocysts.
Heterocysts, yes, for nitrogen fixation. They take nitrogen gas from the air, which most organisms can't use.
And turn it into ammonia, something plants can use? That's huge.
Absolutely vital for ecosystems. They're tiny but foundational.
So, besides the photosynthetic ones, there are also chemosynthetic bacteria.
Yes, chemosynthetic autotrophs. They get energy not from sunlight, but by oxidizing inorganic chemicals, nitrates, ammonia, sulfur compounds.
And they use that energy to build themselves.
Hmm.
Also really important for nutrient cycling, right? Nitrogen, phosphorus, iron.
Crucial roles in keeping those elements moving through the environment.
Okay, and then the biggest group, it seems, are the heterotrophic eubacteria.
By far the most abundant. They rely on consuming organic matter.
These are the decomposers, breaking down dead stuff.
Absolutely essential decomposers, but they also impact us directly in many ways.
Good and bad, I guess. Making yogurt or cheese.
Producing antibiotics.
Helping legumes fix nitrogen in their roots.
All vital, but also causing diseases. Cholera, typhoid, tetanus, even plant diseases like citrus canker.
So, a really mixed bag in terms of human interaction. How do they reproduce, mainly?
Mostly just by splitting into binary fission. very efficient.
Simple and fast.
But under tough conditions, many can form resistant spores, little survival pods.
To wait out the bad times.
Exactly. And they also have ways to swap bits of DNA, a sort of primitive sexual reproduction.
Interesting. And one last group in Monera, Mycoplasma. The odd ones out.
Yeah, they're unusual because they completely lack a cell wall.
Which makes them flexible, I suppose,
Yeah.
and they're tiny.
Smallest known living cells. And they can survive without oxygen. Many are pathogenic in animals and plants.
Wow, so even within Monera, there's surprising diversity.
Definitely.
Okay, let's shift gears to Kingdom Protista, the single-celled eukaryotes, though you said the boundaries are a bit fuzzy.
They can be. As we learn more, especially from genetics, some protists might get moved. But for now, it's this incredibly diverse group, mostly aquatic.
And they're seen as a kind of evolutionary link.
Yeah, linking the simpler prokaryotes in Monera to the other complex eukaryotic kingdoms, fungi, plantae, animalia. Kind of a bridge.
And they're definitely eukaryotes. Proper nucleus, other organelles inside membranes.
Yep. Some have flagella or cilia for moving around, and they reproduce both asexually and sexually cell fusion, making a zygote.
The text mentions several key groups: chrysophytes, diatoms, and golden algae.
Right. Mostly microscopic plankton floating in fresh or marine water, largely photosynthetic.
And the diatoms have those amazing silica cell walls, like tiny glass boxes.
2 overlapping halves, yeah, like a petri dish or a soapbox. And they're incredibly strong, almost indestructible.
Is that where diatomaceous earth comes from?
Exactly. When they die, those silica shells pile up over millions of years, forms deposits we mine.
And used for polishing, filtration.
Uh-huh. It's gritty. So yeah, filters, mild abrasives, maybe even in your toothpaste.
Who knew? And ecologically, they're super important.
Hugely. Diatoms are the chief producers in the oceans, the base of the food web for vast marine ecosystems.
Wow. Okay, next group, dinoflagellates.
Mostly marine, also photosynthetic, come in various colors: yellow, green, brown, blue, red. Depends on their pigments.
And their cell walls have cellulose plates, making them sort of armored.
Stiff cellulose plates, yeah. And they usually have 2 flagella. One runs long ways, the other sits in a groove around the middle, making them spin as they move.
Hence, dinoflagellate, like whirling.
Something like that. And they're famous, or infamous, for causing red tides.
Right, when some species, like Goniolax, bloom like crazy.
Yeah, rapid multiplication. And they can release toxins that are harmful, even deadly, to fish and other marine life.
Nasty stuff. Okay, moving on to euglenoids.
Mostly freshwater, often in stagnant water.
And they don't have a rigid cell wall, they have a pellicle.
Right, a flexible protein layer instead. Gives them more wiggle room. They have 2 flagella, usually one short and one long.
And they have that weird nutritional flexibility.
Very cool feature. In sunlight, they're photosynthetic. But if it's dark, they can switch gears and become heterotrophic, basically hunting smaller organisms.
Best of both worlds.
Yeah.
And their chlorophyll is like higher plants.
Identical pigments, yeah. Suggests a close evolutionary link somewhere back down the line.
Then slime molds. Sound interesting.
They are. They're saprophytic protists, meaning they feed on decaying stuff.
They sort of creep along dead leaves or logs, engulfing organic bits.
Exactly. And under good conditions, they can form this big amoeba-like mass called a plasmodium. It's like one giant cell with many nuclei, and it can spread over a surprising area.
And when things get tough,
The plasmodium changes, forms fruiting bodies that produce spores. These spores have tough walls, very resistant, and get carried off by air currents to find somewhere better.
clever survival strategy. Okay, last group in Protista, the protozoans.
All heterotrophs. They're either predators or parasites, and they're thought to be the sort of primitive relatives of animals.
Makes sense. And they're grouped by how they move.
Largely, yeah. You have the amoeboid protozoans using pseudopodia false feet to move and grab prey, like amoeba itself. Some marine ones have silica shells, and some are parasites, like entamoeba, which causes amoebic dysentery.
Then the flagellated ones.
Use flagella, obviously. It can be free-living or parasitic. Trypanosoma, causing sleeting sickness, is a flagellate parasite.
Ciliated protozoans, covered in little hairs.
Exactly. Cilia beat in coordination to move them around, often quite actively in water. They usually have a gullet or mouth area where cilia sweep food in. Paramecium is the classic example.
And finally, sporozoans.
This group is diverse, but they all have some kind of infectious spore-like stage in their life cycle, and they're all parasitic.
Including the most notorious one.
Plasmodium, the parasite that causes malaria. transmitted by mosquitoes, a huge global health issue.
Definitely. Okay, that covers Protista, very diverse kingdom. Now, let's tackle our final kingdom for today, fungi.
Right. A unique kingdom, all heterotrophic, huge diversity in shape, size, habitat.
We see them all the time, right?
Big.
Molds on bread or oranges.
Mushrooms, toadstools,
Yeast for bread and beer.
parasitic ones causing spots on leaves like on mustard plants.
Some cause diseases in us, too, like athlete's foot or rusts in wheat caused by Puccinia.
True. But they also give us incredibly important things like antibiotics. Penicillin comes from the penicillium mold.
Right, and they're everywhere, cosmopolitan. Air, water, soil, on plants, on animals.
Prefer warm and humid places generally, which is why mold grows faster in the summer, or why we keep food cold.
Most are filamentous, made of threads, except yeast.
Exactly. Yeast are unicellular, but most fungi have these long, thin, thread-like structures called hyphae, singular.hypha.
And a whole network of hyphae is a mycelium. That's like the main body of the fungus.
That's the main vegetative part, yeah. It spreads through whatever the fungus is growing on.
And the hyphae themselves can be different. Conocytic versus septate.
Right. Conocytic hyphae are like continuous tubes filled with cytoplasm and many nuclei, no cross walls.
Like we're in long room.
Pretty much. Septate hyphae have cross walls, or septa, dividing the filament into cell-like compartments, though pores often allow cytoplasm to move between them.
Okay, and their cell walls aren't cellulose like plants.
Nope. Made mainly of chitin, the same tough stuff found in insect exoskeletons and polysaccharides. Big difference from plants.
And how they get nutrition is varied too.
Very. Many are saprophytes, absorbing nutrients from dead organic matter. Essential decomposers.
Others are parasites, living on or in living plants or animals.
And some are symbionts, living in close association with other organisms for mutual benefit.
Like lichens. That's fungi living with algae.
Yep. And mycorrhiza fungi associated with the roots of higher plants, helping them absorb water and nutrients.
Super important relationships. How about reproduction?
Also diverse vegetative methods like just breaking off a piece, fragmentation, or fission, or budding like in yeast.
Simple stuff.
Then asexual reproduction using spores, different kinds like conidia, sporangiospores, zoospores.
And sexual reproduction too.
Yes. Also involving spores, osiospores, ascospores, basidiospores. These are often produced in specific structures called fruiting bodies like a mushroom.
And the sexual cycle involves fusing cells.
Typically involves 3 steps. Plasmogamy, the fusion of cytoplasm from 2 parent cells.
Okay.
Then karyogamy, the fusion of the 2 nuclei. This might happen right away or be delayed.
Delayed.
Yeah, in some groups like ascomycetes and basidiomycetes, you get this intermediate stage called a dicryon, where a cell has 2 separate haploid nuclei, N plus N, for a while before they fuse.
Huh, interesting. And after karyogamy.
After the nuclei fuse to form a diploid zygote nucleus, meiosis happens pretty quickly, reducing the chromosome number back to haploid and producing the sexual spores.
Got it. And the classification within fungi is based on?
Mainly on the morphology of the mycelium, septate or aseptate, how spores are formed, and the types of fruiting bodies they make.
Okay, let's quickly run through the main classes mentioned. Phycomycetes, lower fungi.
Right. Found in water, on decaying wood, damp places. Some are obligate plant parasites. Their mycelium is aseptate and coenocytic, that one long room structure.
Asexual spores made in a sporangium. Motile zoospores or non-motile planospores.
Correct. And sexual reproduction leads to a zygospore. Examples are mucor, Rhizopus the bread mold, and albugo, the parasite on mustard.
Okay. Then Ascomycetes, the sac fungi.
Mostly multicellular, like penicillium, but includes unicellular yeasts like Saccharomyces. Very diverse lifestyles, saprophytes, decomposers, parasites, some even grow on dung.
Coprophilous, right.
Yes, their mycelium is branched and septate the divided rooms.
Asexual spores are conidia, made externally.
On specialized stalks called conidiophores, sexual spores are ascospores, typically 8 of them, formed inside a sac-like structure called an ascus. Asci are often grouped within a fruiting body, an ascocarp.
Examples: Aspergillus, Neurospora for genetics.
Claviceps, which causes ergot, and edible ones like morels and truffles.
Yum. Okay, Basidimycetes, the club fungi.
These are the ones most people picture as fungi mushrooms, puffballs, bracket fungi. Grow in soil, on logs. Some are major plant parasites like rusts and smuts.
Mycelium is branched and septate again.
Yes. Interestingly, asexual spores are generally not found here. Vegetative reproduction by fragmentation is common, though.
So sexual reproduction is key.
Very much so. It often involves fusion of vegetative cells of different mating types, leading to that dikaryotic N+N mycelium. This eventually forms club-shaped structures called basidia.
Ah, the club-in-club fungi.
Exactly. Inside the basidium, karyogamy, nuclear fusion happens, followed immediately by meiosis, producing 4 basidiospores, usually born externally on the basidium.
And the basidia are arranged in fruiting bodies, basidiocarps.
Right, like the cap and gills of a typical mushroom, agaricus, the common mushroom, ustilago, smut, Puccinia rust are examples.
Got it. Last one. Deuteromycetes, the imperfect fungi.
Called imperfect because historically we only knew their asexual or vegetative stages. No known sexual phase.
So if a sexual stage is discovered,
The fungus usually gets reclassified, often moved into the ascomycetes or basidiomycetes, depending on what the sexual structures look like. It's kind of a temporary holding group.
makes sense. So they reproduce only by asexual spores called conidia.
Correct. Mycelium is septate and branched. Many are important decomposers involved in mineral cycling. Some are saprophytes or parasites. Alternaria, Colatotricum, Trichoderma are examples.
Wow, okay. That's a lot to cover in fungi. What an incredible journey, really, from those first basic human classifications.
Right, based purely on use.
So Aristotle, Linnaeus's 2 kingdoms, then Whitaker's 5, and acknowledging it's still evolving.
It really highlights how classification isn't just about sorting, it's about reflecting our deepening understanding of life, especially evolutionary relationships.
Yeah, that phylogenetic aspect seems key now. It's a dynamic process.
Absolutely. Science is always refining, always learning more.
And hopefully, listening to this, you've got a better sense of the unique features and really the vital roles these organisms in Monera, Protista, and fungi play.
From bacteria driving global cycles to the diverse protists connecting different life forms and fungi as decomposers and partners, they're all essential.
So maybe a final thought to leave you with.
Yeah.
With everything changing so fast, especially in fields like genomics, reading DNA, how do you think our classification systems might change next?
That's the big question, isn't it?
Could we discover entirely new categories?
Yeah.
Or redraw the tree of life in ways we can't even imagine right now?
It's entirely possible. As we get more molecular data, we might find unexpected connections or need completely new ways to represent life's diversity. It's definitely something fascinating to think about.